In this video, we're going to talk about the behavior in addictions, specifically how classical conditioning influences the behavior of an addict and how can we change this behavior. We're going to discuss two pairs of key concepts on this topic. The first pair of concepts will be conditioned place preference and cue exposure therapy for addiction. The second pair will be conditioned place aversion and aversion therapy. But first, let's do a quick recap of the physiology of the reward system. Let's start with the definition of addiction. Addiction is a biopsychosocial disorder characterized by compulsive engagement in rewarding stimuli, despite the adverse consequences. Compulsive means the person has an insatiable urge to engage in rewarding stimuli. The word biopsychosocial means addictions have interconnections with biology, psychology, and socio-environmental factors. Addiction affects the reward system in the brain. Here you can see the medial view of the brain. Three main structures of the reward system that you must know are the ventral tegmental area, or VTA for short, which is situated in the midbrain, adjacent to the substantia nigra, the nucleus accumbens, and the prefrontal cortex. Engaging in enjoyable activities generates action potentials in the dopamine-producing neurons of the ventral tegmental area. In our example, the boy is eating. When you're eating, the main three things that you're enjoying are the taste of food, the sight of food, and the smell of food. The parts of the brain responsible for sight, taste, and smell, respectively, will eventually stimulate the VTA. It's not a direct stimulation, but through a chain of neurons. The ventral tegmental area contains a lot of neurons that produce and release dopamine, called dopaminergic neurons. These neurons will send axons to the nucleus accumbens, which is a component of the limbic system, and to the prefrontal cortex. The pathway that connects the ventral tegmental area to the prefrontal cortex is called the mesocortical pathway. The pathway that connects the ventral tegmental area to the nucleus accumbens is called the mesolimbic pathway, also known as the reward pathway. Let's zoom in on these structures. Here you can see the VTA, the nucleus accumbens, and the mesolimbic pathway. Now imagine you're eating your favorite food. The parts of the brain responsible for sight, taste, and smell will stimulate the amygdala, which stimulates other neurons, which, in turn, will stimulate the dopaminergic neurons from the VTA. The dopaminergic neurons will release dopamine, which will bind to the receptors on the medium spiny neurons and will inhibit them. The inhibition happens due to the D2-like receptors. They are coupled to the G inhibitory protein, which will inhibit the neuron. So now, the GABAergic neurons are inhibited. Because of that, they will release little to no GABA. Normally, GABA binds to GABA type A receptors. Chloride ions enter the neuron and hyperpolarize it, thus inhibiting it. But since there's little to no GABA in the synaptic cleft, very few chloride ions are going to enter the cell. If GABA doesn't inhibit the neurons from the parts of the brain responsible for pleasure anymore, this is called disinhibition. Or you could say stimulation. This stimulation creates the experience of pleasure. Now, we have to see where in the brain is this red neuron actually situated. In order to do that, firstly, let's take a look at the coronal plane of the brain. Here you can see the putamen, the dorsal pallidum, which is commonly known as the globus pallidus and consists of the external and internal segments. Then we have the ventral pallidum, the thalamus, and the prefrontal cortex. We'll start with the medium spiny neurons. The medium spiny neurons of the nucleus accumbens will send axonal projections to the basal ganglia and the ventral analog of the globus pallidus, known as the ventral pallidum, which is a major center of reward in the brain. The ventral pallidum, in turn, projects to the medial dorsal nucleus of the dorsal thalamus, which sends neurons to the prefrontal cortex. The neuron in black is one medium spiny neuron from the nucleus accumbens. When this neuron is inhibited, it releases little to no GABA. As a result, the neurons in red, which are responsible for pleasure, are stimulated, and they release glutamate. Glutamate serves as the major excitatory neurotransmitter in the vertebrate central nervous system and also is the main neurotransmitter of communication between the thalamus and the cerebral cortex. So, one more time, those two neurons in red are responsible for pleasure. Now let's look at the same coronal slice once more. Speaking about addictions and memory, there is a connection between the prefrontal cortex and the hippocampus. Pay attention to these dark blue neurons. These neurons are particularly important for memorizing details about the rewarding stimuli. Behavior in addictions depends on the memory of the rewarding stimuli and the memory of the rewarding experience. This is the main focus of this video. So pay close attention. Because of the connection between the prefrontal cortex and the hippocampus, an addict can form associations between the memory of the rewarding experience of drug use and the memory of the cues in the environment. For example, the aspect of the room where he took his last dose or the aspect and mood of his drinking buddies. As it turns out, the cues in the environment are strong mediators of the drug-seeking behavior and can trigger relapse even after months or years of abstinence, just like in this scene from BoJack Horseman.
Goji berry, flax, chia, shark fin, and white rhino horn. Sober, so good. BoJack?
Hey, Sarah Lynn. You want a party?
Oh, thank God. Yes!
Okay, for those of you who are still confused, in this scene, Sarah Lynn is the addict, alcohol is the rewarding stimulus, BoJack represents the cue in the environment because he's Sarah Lynn's old drinking buddy. This cue in the environment is responsible for triggering alcohol-seeking behavior and relapse after 9 months of abstinence. This whole interconnection between the rewarding stimulus, the memory of the rewarding experience caused by the rewarding stimulus, and the memory of the cues in the environment related to the rewarding stimulus, are components of classical conditioning, also known as Pavlovian conditioning, which was named in the honor of the Russian physiologist, Ivan Pavlov. Do you remember Pavlov's dog experiment? Pavlov rang a bell, then fed the dog. In this case, the food is the rewarding stimulus, the pleasure and excitement from the food is the rewarding experience, and the noise of the bell is the cue in the environment. Now, it's pretty hard to grade the level of pleasure and excitement in dogs. On the other hand, the amount of saliva produced by the dog in response to food is kind of a measure of pleasure and excitement from food. The secretion of saliva is a part of the food-seeking behavior. The food is an unconditioned stimulus because it naturally elicits salivation in dogs. So, salivation, in this case, is the unconditioned response to food. After a short period of time, Pavlov rang the bell, then fed the dog again. Initially, the noise of the bell was a neutral stimulus because, on its own, it didn't elicit salivation. But since the dog heard the bell sound every time he was fed, the bell sound was paired with the presence of food. But what happens inside the brain while the pairing of the stimuli takes place? Inside the brain, the pairing of the stimuli is actually the pairing between the memory of the sound of the bell and the memory of the presence of food while feeling pleasure. Long-term memory is stored in the cerebral cortex. Now back to our experiment. Eventually, when the dog heard only the bell sound, it salivated, as if it was about to receive food. Since now the dog responds to the noise of the bell in the same way it responds to food, the noise of the bell is no longer a neutral stimulus. It's a conditioned stimulus. So, to generalize, conditioning is a form of learning in which a stimulus becomes increasingly effective in evoking a response. So, basically, a conditioned stimulus means a learned stimulus. In our case, the noise of the bell becomes increasingly effective in evoking salivation. And when something responds to a conditioned stimulus, that response is referred to as a conditioned response. Now for the juicy part. If we were to compare Pavlov's dog experiment to the behavior in drug addictions and behavioral addictions, we would observe the following. Instead of the dog we'll have the drug addict, or the addict in general, instead of the food we'll have the addictive drug, or the addictive behavior, and instead of the noise of the bell we'll have the cues in the environment, for example, the aspect of the room where the addict took his last dose, the mood of his drinking buddies or activities that were often done while using drugs. Emotions like stress, anxiety, boredom, and depression can all be triggers for drug use. Even positive emotions, such as happiness or excitement, can be triggers if they are associated with past drug use. These emotions are triggers and also cues in the environment. Full disclosure, the drug addict doesn't salivate when on drugs. But he feels pleasure, euphoria, in other words, the rewarding experience. Let's say the room is the cue in the environment. Initially, the aspect of the room was a neutral stimulus because, on its own, it didn't elicit the drug-seeking behavior. But since the addict saw the same room every time he took the drugs, the aspect of the room was paired with the presence of the addictive drug while feeling pleasure. The cues in the environment are initially neutral stimuli, but over time they become conditioned stimuli. Now, when the addict enters the same room, it can elicit the drug-seeking behavior. The same goes for our example. Sarah Lynn is the addict, the alcohol is the addictive substance, and Bojack is a drinking buddy of Sarah Lynn, or, scientifically speaking, a cue in the environment. Initially, the presence of Bojack was a neutral stimulus, meaning it didn't trigger drug-seeking behavior. But since Sarah Lynn saw him again and again while taking addictive drugs, the presence of Bojack was paired with the presence of the addictive drug while feeling pleasure. So, over time, the presence of Bojack becomes a conditioned stimulus. Now, if Sarah Lynn sees Bojack again, this can elicit the drug-seeking behavior. The analog to the drug-seeking behavior is the food-seeking behavior in dogs. And I am talking about our examples because a dog can be a drug addict too. The pleasure from the food and the pleasure from the drug consumption are both rewarding experiences. People always seek rewarding experiences. Think about it. We always do something that's rewarding. Even when it's not directly rewarding, it's indirectly rewarding. You know, like, doing something that you're not really into because achieving the goal is rewarding. Even when the action doesn't look like it's rewarding, something is rewarding behind it. Evolutionarily speaking, seeking rewards helped our ancestors survive. Finding food, avoiding danger, and nurturing relationships all provided rewards that ensured our survival and procreation. You know what's funny? If you think about it, bacteria and other organisms without a CNS don't need a reward system to keep them alive, they just go with the flow, but the more complex life forms like humans would disintegrate into atoms without one. Now back to addiction. Why does the addict keep seeking the rewarding experience? There's a memory-related reason for that. Think about this example. The drug addict felt good in the past after he had done drugs. To this moment, he remembers that insane amount of pleasure. He can remember that, thanks to the connection between the prefrontal cortex and the hippocampus that we previously discussed. He wants to feel good again. I mean, who doesn't? The short-term memories of the rewarding experience are initially formed and stored in the hippocampus, before being gradually transferred to long-term storage in the neocortex and disappearing from the hippocampus. In the end, these memories can kind of screw us over, because they are the origin of the drug-seeking behavior. You remember how it felt. You want to feel it again, and a lot of drug addicts would do almost anything to feel it again. The magical trap of addiction has snapped shut. The same mechanism goes for any other drug addictions and behavioral addictions. Of course, doing cocaine is much more addictive than watching Netflix. Still, both activities are addictive. Another example of classical conditioning used in research is conditioned place preference. If we were to compare Pavlov's dog experiment to the conditioned place preference, we would observe the following. Instead of a dog we'll have, usually, a mouse. Instead of food we'll have an addictive drug, like heroin. And instead of the noise of the bell we'll have the aspect of the chamber in which the mouse is located. For this experiment, we need two chambers, numbered 1 and 2. They are connected by a small neutral chamber. Each compartment has a different cue in the environment. Cues can be visual, for example, stripes on the wall in the first chamber, and stars in the second one. We can have tactile cues, for example, the texture of the floor. In the first chamber the floor is made out of wood. The second one is made out of glass. Cues can also be olfactory. For example, in the first room we have the scent of cinnamon, in the second room the scent of roses. The mice are administered a drug, for example, heroin, only when they are in chamber number 1, and saline solution when they are in chamber number 2. Initially, the stripes on the wall in chamber number 1 are a neutral stimulus because, on their own, they don't elicit the drug-seeking behavior. But since the mouse sees the same stripes on the wall, let's say, at least 5 times, when he is injected with drugs, the stripes on the wall become paired to the presence of heroin while feeling pleasure. So again, we're pairing a neutral stimulus to an unconditioned stimulus, which is the presence of heroin. The stripes on the wall have just become a conditioned stimulus. After a couple of days, the rodent will prefer the first chamber and will tend to avoid the second one. It spends much more time in the first chamber than in the second one, even without being given more heroin or saline solution. By measuring the amount of time an animal spends in an area associated with an unconditioned stimulus, for example, heroin, the researchers can say if the mouse likes the unconditioned stimulus or not. Our mouse sure likes heroin. Look, 23 hours in the first chamber versus 1 hour in the second. So, to summarize, conditioned place preference is used to measure how much an animal likes a particular stimulus, which is usually a drug. This method is about a place preference, meaning which place does the animal prefer. It's important to understand and remember the term conditioned place preference because it's often mentioned in studies. As an example, let's take a look at this study about amphetamine-induced place preference in humans. This study concludes that humans, like non-humans, prefer a place associated with amphetamine administration. These findings support the idea that subjective responses to a drug contribute to its ability to establish place conditioning. This study is just one example. Classical conditioning and conditioned place preference have some helpful recovery implications. Research has demonstrated that if we ring the bell many times, without food, the paired association ends. Eventually, the bell will no longer elicit salivation. Let's return to our previous example. Quick recap. Sarah Lynn is the addict, the alcohol is the addictive substance, and Bojack is a drinking buddy of Sarah Lynn, or, scientifically speaking, a cue in the environment. Initially, the presence of Bojack was a neutral stimulus, meaning it didn't trigger drug-seeking behavior. But since Sarah Lynn saw him a lot of times, previously, when she took drugs, the presence of Bojack was paired with the presence of the addictive drug while feeling pleasure. So, over time, the presence of Bojack becomes a conditioned stimulus. Now, if Sarah Lynn sees Bojack again, this can elicit the drug-seeking behavior. Let's imagine a different scenario. If Sarah Lynn saw Bojack repeatedly and she wasn't drinking at the same time, the cravings for alcohol, which are related to Bojack, would eventually subside. But why? Because if the addict gets exposed to the cues in the environment, without taking the drug, the paired association subsides. See? No pairing anymore. So initially, the presence of Bojack is a neutral stimulus. Then it becomes a conditioned stimulus because it can trigger the drug-seeking behavior. If Sarah Lynn saw Bojack repeatedly, without engaging in addictive behaviors, his presence would lose the power to induce cravings because the presence of Bojack would become a neutral stimulus again, as in the beginning. This is the mechanism of the cue exposure therapy. It's called cue exposure therapy because we're exposing the patient to the cues in the environment that can elicit the drug-seeking behavior, without taking the drug. This is one way in which therapists use classical conditioning to diminish and eventually eliminate the addictive behaviors. Now let's analyze the opposite situation to conditioned place preference, called conditioned place aversion. Aversion means a strong dislike. Let's have the second experiment using a toxic salt called lithium chloride. The mouse is given lithium chloride solution in the first chamber and saline solution in the second chamber. Lithium chloride will cause nausea and disgust. The mouse will avoid the first chamber and will spend much more time in the second one because the pairing between the stimuli takes place. Specifically, the neutral stimulus, which consists of the stripes on the wall in the first chamber, is linked to the unconditioned stimulus, which is the presence of lithium chloride that naturally induces disgust and nausea. After a couple of lithium chloride injections, the stripes on the wall will become a conditioned stimulus because they will be able, on their own, to bring back memories of disgust. That's why the animal will avoid the first chamber. Aversion therapy is one application of conditioned place aversion. In aversion therapy, we intentionally form a paired association between an unwanted behavior and an unpleasant experience. Let's just say, for the experiment's sake, that the unwanted behavior is the mouse entering this chamber. In the case of an alcoholic, the unwanted behavior is drinking alcohol. We can administer a drug that causes someone to become horribly nauseous and even vomit, if he or she ingests even the slightest bit of alcohol. This drug would be the analog of lithium chloride, not by the mechanism of action, but because they both create an unpleasant experience. The administration of such drugs forms a paired association between alcohol intake and the feeling of nausea and disgust. Prior to the aversion therapy, a person would ordinarily associate alcohol intake with positive feelings. After aversion therapy, alcohol is associated with nausea and vomiting. For many aversion therapy patients, even the thought of drinking alcohol elicits feelings of nausea. The effect of aversion therapy can wear off over several months. However, during the period it is effective, a person can learn to develop a new manner of healthy living. They can practice coping skills that strengthen their ability to enjoy life without alcohol. To clarify, although drinking is a voluntary behavior, cravings and desire for alcohol are not. There are other forms of aversion therapy which can be used in cocaine addiction, cigarette addiction, just to name a few. Their mechanisms are similar to the one described above. In conclusion, aversion therapy diminishes the craving and desire to perform a certain behavior by diminishing its appeal. As a quick recap, conditioned place preference measures an animal's preference for a place associated with a drug. Cue exposure therapy aims to extinguish drug-seeking behavior by exposing addicts to cues without the drug. Conditioned place aversion, the opposite, creates a dislike for a place associated with an unpleasant experience, like nausea induced by lithium chloride. Aversion therapy applies this concept by pairing unwanted behavior with unpleasant experiences, diminishing its appeal and cravings.